Let's dive into a technology that sounds like it's straight out of science fiction. The idea of creating a baby with, technically, three biological parents. It's a real procedure, and it was designed to prevent some of the most devastating genetic diseases out there. But, as you can imagine, it also forces us to ask some really profound questions about science, ethics, and our future.
So, right off the bat, let's just ask it. Could a baby really have three biological parents? It seems impossible, right? Well, the answer is sort of. Yeah. So, let's break down what that actually means, the absolutely heartbreaking problem this technology aims to solve, and the incredible science that makes it all possible. Okay, so before we get to the amazing solution, we really have to understand the problem, and it's a brutal one. It's a kind of cruel genetic lottery known as mitochondrial disease. Inside pretty much every single cell in your body, you have these tiny little structures called mitochondria. You can think of them as the power plants for your cells, generating almost all the energy you need to, live. But what happens when those power plants are faulty because of mutations in their DNA? Well, that's mitochondrial disease. And here's the absolute key. That mitochondrial DNA, it's inherited exclusively from your mother. And the impact, wow, it's just devastating. Because mitochondria power our most energy-hungry organs, like our brain, our heart, our muscles. Any failure can lead to catastrophic problems. We're talking brain damage, heart failure, muscle weakness. These are progressive, debilitating conditions that, all too often, are completely incurable. And this isn't just some abstract number. 1 in 4,000 children born in the U.S. has a mitochondrial disease. That means for thousands of families, the dream of having a biological child comes with this terrible, terrible gamble, the risk of passing on a disease with no cure, at least until now. So how do you solve a problem that's literally written into our cells? Well, scientists came up with a truly revolutionary solution. Let's get into the science of it, and while we're at it, let's demystify that controversial three-parent label. This chart right here really shows you just how misleading that three-parent label is. Look at this. Over 99.9% of a baby's DNA, the stuff that determines traits like hair color, height, and so on, comes from the mother and father. The donor's contribution is a tiny, tiny fraction, just 0.1%. That's only the 37 genes needed to run the mitochondria, not the 20,000 plus genes in the nucleus that make us who we are.
Here's a great way to think about it. It's like you have a laptop with a faulty battery. The procedure doesn't touch the hard drive. All the core data, the operating system, your files, everything that makes the computer yours is kept completely intact. All that's getting swapped out is the defective battery pack. The child's core genetic identity is completely unchanged.
So, how in the world do they perform this microscopic swap? It's pretty amazing. Let's walk through one of the main techniques, which is called maternal spindle transfer, or MST for short. It's a super delicate 4-step dance. First, scientists take the mother's egg and carefully remove the nucleus. That's where her core genetic code is. Second, they take a healthy egg from a donor and remove its nucleus, leaving behind the healthy mitochondria. Third, and this is the magic part, they place the mother's nucleus into the donor's empty egg. And just like that, you have a reconstructed egg with the mother's DNA and a healthy set of mitochondria, which is then fertilized by the father's sperm. Now, a procedure this revolutionary doesn't just appear out of thin air. It was built on years and years of careful research, and a really critical chapter in that story involves some of our closest relatives, non-human primates. As Dr. Shukrat Matalapov's team at Oregon Health and Science University put it, their work simply would not have been possible without first trying and perfecting the technique in primates. This research was absolutely essential to prove that the whole concept wasn't just possible, but that it could be done safely. And prove it? They did. The research was a huge success. It resulted in the birth of 3 healthy macaque infants, the first ever primates born using this incredible technique. But, the most critical part of this research wasn't just the birth. It was the long-term validation. These monkeys weren't just healthy when they were born. They developed completely normally and stayed healthy for over 3 years with no negative side effects. This was the crucial safety data that was needed before anyone could even think about trying this with humans. Okay, so understanding the science is one thing, but this technology takes us into a totally uncharted ethical frontier. We have to weigh its incredible promise against some very real, very complex perils. On one hand, the promise is just immense, preventing horrific, incurable diseases and allowing families to have healthy children that are genetically theirs. But on the other hand, the peril is also significant. This is what's known as a heritable genetic modification, or a change to the human germline. That means the change doesn't just affect one person, it gets passed down through all future generations. And while the animal studies look good, the long-term effects in humans, well, they're still unknown, and that raises all kinds of questions about identity and what it means to be a parent.
So, with all this ethical complexity, it's no surprise that different countries are taking very different approaches. The UK, after a lot of review and public debate, actually became the first to approve the technique. The US, however, has restricted it, saying more research is needed and putting up some major regulatory hurdles.
And I really want to stress this point. This whole debate is no longer theoretical. We're not talking about some far-off future. The technology is already here. Babies have been born using these techniques in the UK, in Mexico, and other places, which moves this entire conversation out of the lab and squarely into the real world. And that reality leaves us with this final, fundamental question. This technology was created with one goal, to cure disease. But it opens a door to modifying the human genetic code in a way that gets passed down forever. So where do we, as a society, draw the line between curing disease and, well, designing our children? That's a question we are only just beginning to answer.